inputs given. The next guy I have to introduce, it's a pleasure to see Gerrit Borchard. He's a professor at the University of Geneva from Switzerland, but he's not a real Swiss guy. We imported him from outside, but nevertheless...
Actually, actually the US. Thank you, Stefan.
He will introduce himself in the topic he will address, because I think there, again, the compendial issues is a topic we have seen so far as well in having the views. Floor is yours.
Thank you, Stefan. I'm also a member of the steering committee of the NBCD working group. As you said, I am, since more than 10 years, I'm one of the European, or shall I say, Swiss scientists involved in nanomedicine research, and based for more than 10 years in beautiful city of Geneva. And this, of course, is the view from my office in Geneva. I'm also a trained pharmacist, so that's why dealing with pharmacopoeial issues comes kind of natural to me. So I'm going to introduce what initiatives are currently ongoing, or some of the initiatives that are currently ongoing at the USP and the European Pharmacopoeia. Basically, just to give you an introduction, there are different, business models for the three major pharmacopoeias. You have the Japanese Pharmacopoeia, which is part of a governmental body. You have the European Pharmacopoeia, which is kind of intergovernmental, as a part of the European Directorate of the Quality of Medicine, so it's a European directorate. And you have the US Pharmacopoeia, which is a not-for-profit organization which was incorporated in 1900. So it's independent of the governmental organizations. Now, for the EDQM, that's the Council of Europe Directorate. It's based on a convention in 1964. Its mission is, as is for all the pharmacopoeias, the access to a basic human right, access to good medicines, quality medicines and healthcare. In 1994, the EU signed the European Pharmacopoeia Convention for the procedure of certification of suitability of certain monographs, and during the years after that, there was transfer of activities for different products also to the EDQM. Now, the obvious task of pharmacopoeias is to protect public health. There's one common compulsory standard, which is mandatory for the member states of the European Pharmacopoeia. The European Pharmacopoeia is mandatory at the same date in 37 member states, and the European Union, -1. It's legally binding. It has quality standards for all medicinal products, raw materials, preparations, and so forth. And of course, there is an international exchange of ideas, which is rather slow going, but it does exist, the harmonization between the European Pharmacopoeia and their sister pharmacopoeias in the US and Japan. Now, just to give you an idea how this is done, I've been involved in the EDQM for a couple of years now, and I've seen it with my own eyes. So basically what happens if somebody has an idea for creating a new monograph, there's the stakeholder, most of the time a national organization like Swissmedic or another organization from the member states, giving a proposal to the European Commission, COM. The COM then decides whether or not to take this onto the working program. They elect a group of experts or a working party. I'm a member of a working party, which sounds more fun than it really is. And this working party then tries to develop a first draft of the monograph. The monograph is then, or this first draft is then published in Pharma Europa for comments by stakeholders, so you can get back the comments, and we try as a working party to implement these or not into the draft. Then this is resubmitted. National pharmacopoeia authorities process the comments and received on the graph. Then the European Pharmacopoeia compiles all this. It goes back to the working party or group of experts, and then it's finally submitted to the COM, which then or not adopts the monograph, and it's published again about 6 months later. And this is not carved in stone. The monographs in the European Pharmacopoeia are not carved in stone, but there's an exchange of information, a revision that's constantly taken place between stakeholders and the European Pharmacopoeia. Now, there's the NBC Working Party, which was created already in June 2011 by an initiative by Swissmedic. I got into the whole business in, I think, 2013. Stefan recruited me to this enterprise. It was a decision of the European Pharmacopoeia Commission to add on its working program as a first elaboration for a monograph of NBCs, iron sucrose concentrated solution, as a first example for non-biological complexes. And that was intended to be followed up by other NBCs, or other complex drugs. This is the iron sucrose product. It's basically built like an M&M, if you want. You have a core of iron oxyhydroxide and a layer, several layers of sucrose around that. Looks easy, but it's not. In my second job at the University of Geneva, we are doing research on that in order to do the PCC, the physical-chemical characterization of these products, and we see differences in different products for iron sucrose, just to tell you that. The working party, just to give you an idea who's working in there, so we have people from academia, we have people from regulatory authorities like the BfArM in Germany and the AEMPS from Spain, but also representatives from companies that do have products on the market in this iron sucrose business, and they are both originators and generic companies that come together and try to work on this. Now, this work is pretty tough. We're currently discussing something to give you an idea. We're discussing one of the analytical methods we have to use in order to characterize these products is polarography. On the other hand, the EDQM is trying to ban the use of mercury in their monographs. So what you do, can you find an alternative method in order to do that? The other thing is that we're currently discussing the possibility to create a chemical reference standard for iron sucrose. And as Scott just mentioned, I don't know. I don't know how this CRS would look like in the end for this particular complex drug. Now, if you now switch across the Atlantic, it was very interesting. I visited the USP a couple months ago and talked a little bit with them how their monographs are made. It's a different approach. Here, it's not a national authority, but it's basically the sponsor who comes to the USP and suggests or submits a proposal for their product. That can be originator companies, but more and more they see also generic companies. That's been reviewed by their council of experts. Then they then they have an approval publication of Pharmaceutical Forum for public comments, and the expert committee then reviews these comments, and with the USP scientific liaison is obviously also involved in this. Now, the USP scientific liaison compiles and analyzes this complex comments and gives the USP expert committee a response, and there are two possibilities. Either a further revision is needed, so it goes back again to this review process, or if everybody agrees that there is no further revision needed, the revision proposal becomes official, it's published in the USP NF, and so forth. Now, currently ongoing at the USP is a discussion about Glatiramers. You may have heard of it, Copaxone, generic Copaxone. Just to give you a background, a little bit of science in this talk, is that the Glatiramers, or the Copaxone, was actually found by scientists, researchers at the Weizmann Institute in the 60s. They basically wanted to create an animal model for multiple sclerosis by creating an experimental autoimmune encephalitis in these guinea pigs. Now what they did, they chopped up this myelin basic protein that insulates neurons against the environment. They chopped it up into amino acids, and they put these amino acids back together again to form polypeptides. And then they injected these polypeptides as synthetic copolymers into the guinea pigs, thinking that this would create an autoimmune disease and an EAE. However, they saw that actually these guinea pigs were resistant against EAE. They were protected against it, and they lived happy ever after. Now, this Copaxone, so Teva picked this up, and they developed the Copaxone from this, and there are 10 to the 26 possible different peptide structures. It's not only a complicated drug, it's a complex drug, if you would agree. The mechanism of activity is unknown because you cannot identify single APIs, single peptide structures that do the trick, so you cannot also establish a PK/PD profile. However, in April 2015, the FDA approved it through the generic pathway, the Sandoz momentous Galtopa, and today there are colleagues of mine who are involved in the USP expert panel that has been established in 2016 to develop a monograph on these very complex drugs. Together with stakeholders from Teva, from different companies that are either participating or functioning as observers there. Last but not least, I said this earlier, there's a pharmacopoeial discussion group which was set up in 1990 where you have this harmonization between the major pharmacopoeias. This is to discuss what is going on in the different pharmacopoeias to avoid redundancy in testing, so to acknowledge the monographs from the other pharmacopoeias in order to reduce overall cost, but also reduce time required for the medicines to be made available to the patients. There's also a harmonization ongoing with other organizations like ICH in these activities. And I think in order to put this puzzle for the PCC and the puzzle of nanomedicines all together again, we need all the king's horses. Thank you very much.
Thank you. Questions for Gerrit? I have.
Go ahead.
I'm very intrigued by this work, and I'm just asking whether in your group, for example, for Copaxone, where you have 126 forms, whether there have been new thinking on how to characterize the mixture, because we learn from other fields. That sometimes the most important thing is the proportion between the various components. So it's like a sort of the need of bioinformatic methods to look into the various components, how they combine each other. And the other thing, the second point is that I've been made aware of some methodologies in using macrofluidic and RNA expression with cells to characterize the functioning of the mixture for characterizing the mixture. You cannot do the individual bits. You don't have a fingerprint, it's too many. But then you may, I don't know whether you are aware and if you discuss in these groups this kind of novel approaches to characterization of mixtures and complex mixtures, because we have been confronted with these discussions in the Innovation Task Force.
So I am not a member of this expert panel. I may have observational status there, but very much involved in the iron sucrose. But just to answer your question, I'm looking at Vera. Vera and her colleagues from Teva blew me away of the analytics they are using for that. And I must correct you, this is 10 to the 26 different structures that can be. This is, I think, 1,000 times more than there are stars in the known universe. That's how complex that is, even though FDA said, no, you can characterize this mixture, you can see from MS spectra or whatever, that this is the same, so they accepted it. That's what I got from the reasoning of the FDA.
Thanks. Any other questions? I had a quick question. Pharmacopoeias, and in particular the European Pharmacopoeia. What is your position with respect to the use of animals in the monograph for the testing of these complex drugs? Do you consider them?
No.
There,
this is a very rare occasion that we would use animal studies for that. When it comes to like testing immunogenicity, you know that the limitless test or something like this that's been used, but for now we did not consider at all animal studies. What we did consider in the beginning, but that's been a tough discussion, is to look at in vitro tests like macrophage uptake studies for iron sucrose and these products. It's the immune system is highly involved in the PK/PD. So we were thinking of, establishing an in vitro study for that. But animal studies, no.
Okay.